thank the organizers for giving me a chance to speak, even though my voice is not quite what it normally is. If I sound a little incoherent, I think I have not RSV, but I think I have a little bit of a rhinovirus infection right now. So we've been interested in the functions of the non-structural proteins of RSV for a long time. This is actually sort of a love-hate relationship I've had with the non-structural proteins since I was a fellow in Peter Collins' lab. Just to remind you, the non-structural proteins are the first two genes in the gene order, so they're the promoter proximal genes. They're also called non-structural proteins because they are not located in the virion themselves, as opposed to all these other proteins, as we've heard, the G protein, F protein, and M protein, all make up part of the virions, while the non-structural proteins are only expressed in the infected cells. The initial things that I did when I first got into Peter's lab was to look at what the functions of the non-structural proteins did. And one of the obvious things to do was to take them out of the genome with Peter's reverse genetic systems, which was just developed. One of the things we were able to do at the time was to test these viruses in chimpanzees. And what you can see is that when you knock out the NS1 protein, you get a virus that is highly attenuated, still immunogenic, even at a 10-fold higher dose than wild-type RSV in a chimpanzee. And as you can see, also another virus that is of interest, this Delta MT2, which I think Ula will be talking about in a couple days, is also very attenuated and immunogenic. So out of these studies came a couple potential vaccine candidate for the live attenuated viruses. Well, so we were focusing on describing what NS1 does, and one of the things that we know that has to happen during a viral infection is that when a virus comes in, it stimulates the interferon response. And over the course of a number of years, people have looked at both NS1 and NS2 and found that it has both an effect on the induction of interferons, so blocking the production of interferons, as well as blocking the signal transduction from the interferon receptor. What I'm going to focus on today is really this idea that RSV that lacks the NS1 protein can actually no longer block the induction of interferons. The reason we're kind of interested in these interferons and the type 1 interferons is because a lot of evidence more recently has been showing that the production of type 1 interferons is essential for immunogenicity. In this slide, you can see that induction of type 1 interferons allows for maturation of dendritic cells from both the macrophage and immature dendritic cell lines to produce myeloid dendritic cells that will stimulate both CD4 and CD8 cells to make CTL and Th1 cells. There's also evidence from measles virus that type 1 interferons are important in inducing robust humoral immunity as well. So the idea then is if you can block the ability of RSV to antagonize the interferon response, you could potentially get a more immunogenic virus. Okay, so work from Ellen Brazier's group showed basically that there are two separate pathways that are stimulated by RSV when it induces interferons in infected cells. The early response is due to the Rig-like helicase pathway. So the Rig-like helicases are dead box helicases which have an RNA helicase which recognizes 5' triphosphorylated RNAs, and it stimulates a number of different downstream molecules resulting in the phosphorylation of a transcription factor called interferon regulatory factor 3, which then dimerizes when it's phosphorylated, translocates to the nucleus, and then stimulates transcription from the interferon beta gene. And so what we've done to look at this is we've developed promoter assays where you basically look at either interferon beta promoter driving luciferase or just an ISG56 promoter, which has an ERF3 binding site, and that drives luciferase. So what you can do then is to take different molecules within this pathway. You could take the N-terminal part of the helicase and stimulate basically transcription from either the interferon beta promoter or the ISG56 promoter. So what we found is that the NS1 protein, when you add it in, transfect it in with any of these components of the interferon, the Rig-like helicase pathway, so you can block the ability of these components to stimulate either ISG56 promoter or interferon beta promoter activity. And this also works for a molecule in the downstream interferon pathway, the TLR3 pathway, which can be stimulated through transfection of the TRIF molecule. So with this assay, then you can actually look at the ability of NS1 and where it blocks the different levels of the interferon induction pathway. The problem for us is that it seems to block a number of different things. So we and others have found that NS1 seems to localize with the mitochondrial kind of activating gene on the mitochondria. Solenberg's group has found that NS1 causes proteasomal degradation of some of these products like IKK epsilon and TRAF3. And a report from Xiaoyong Bao's lab showed that NS1 can actually act in the nucleus and block transcription of particular RNA polymerase II-driven transcription. So we're still not quite sure where NS1 acts, but we know that NS1 is very effective at blocking the production of interferons. So the problem with the NS1 knockout virus then is it's great at producing interferons, but it also has an inability to replicate well, even in an interferon-deficient environment. So you can see here, for example, that in A549 cells, which have interferons, the virus doesn't grow very well. But even in viro cells, which lack type 1 interferon genes, it still doesn't grow very well, even though it induces significant amounts of nuclear translocation of ERF3 or interferon. So this gave us our hypothesis then that we could actually separate these two activities so that the replication activity of NS1 is separate from its interferon antagonism activity. And then when you take these two apart, then you can develop a virus that will replicate pretty well, but will also induce a lot of interferons so that it can be more immunogenic. This is a nice hypothesis, but how do you go about doing it? Not being a biochemist, I used to look at proteins in domain form. You've seen from Jose and Mark that F and G have domains. We tried to do some basically wreck and check, basically delete domains of NS1, and basically nothing happened. It just made the protein completely unstable. We tried some really fancy bioinformatics, and that didn't work either. Focusing on these kind of conserved regions between the different strains of bovine RSV. So finally, when I got to USF a few years ago, we hooked up with a guy who works on intrinsic disorder. So in intrinsic disorder predictions, you look at proteins and you look at the order or disorder of the protein sequence. The more disorder, that region hypothetically has more function where it can actually bind to different proteins and have different conformations. The more order, the less likely it is to bind to multiple proteins. And so using intrinsic disorder predictions, we found that the termini of the NS1 protein seem to have a little bit more disorder. There's a little bit of a bump here in the middle as well. So then we set about trying to look at those particular regions of the NS1 protein to see whether they were involved in either the replication or the interferon antagonism functions of NS1. What we found is that we could do 10 amino acid deletions without too much problem. The 10 amino acid deletion proteins were a little bit unstable. This is transfected cells. You could stabilize them with the production of NS2. And then we did our ISG56 activation assay, and we could find that the deletions of the N-terminus and the C-terminus seem to allow for interferon transcription, whereas some in the middle do not. These guys, we expect, it's because they are a little bit less stable that they don't really block interferon activation as well. But we focused on these guys at the N and C termini. We put these into a recombinant virus using Peter's reverse genetic system. And we could see then with the reverse genetics, with the recombinant viruses, if you infect A549 cells, now these ones that have deletions in the N and C termini seem to produce a lot more interferon beta. This is qPCR assay. Okay, so this was great, but then we looked at replication, and I've separated them out, the viruses out into viruses that induce interferons and the ones that don't. You can see that there are regions in the middle of the virus that seem to be important for replication because these don't replicate, even though they don't induce interferons. But the ones that do induce interferons, we can really never get wild-type like replication. We could get somewhat intermediate levels, about a little bit less than a log final titer higher. So to make a long story short, we went through a whole series of mutations, making smaller and smaller deletions. We came up with a region between amino acids 5 and 15 in the N-terminus, and we made single amino acid deletions in that region. We did the transfection assay. We found a couple amino acids and a couple combinations that actually seemed to be involved in blocking interferon activation. So then we took those, put them into the virus, and now you can see some of them, actually some of the ones that didn't look like they did much within the transfection assay actually have activity. So they produce about 10-fold more interferon mRNA in infected cells. But a couple of the ones that we really expected to be from the transfection assay, this deletion of the 11th amino acid, deletion of the 9 and 12, they actually seem to have a lot better or less ability to block the interferon production. So you have a lot more interferon message produced. Unfortunately, these didn't have full replication activity, but it has a little bit higher replication activity than the 10 amino acid deletions. You can see some of them that they're all a little bit different, but many of them have this kind of intermediate phenotype. So now we have at least an idea that we can separate out some of the replication functions from the interferon antagonism. So to basically summarize, what we found is that NS1 seems to have these both interferon antagonism and replication functions, that it seems to be localized to the N and C termini. We haven't really checked much of the C terminus yet, but that's what we're going to do next to see if we can balance the replication and interferon production activities. And we've shown that we can delete single amino acids and still have this segregation between the interferon antagonism and the replication activities. Okay, so to finish off, I'd just like to thank the people that did most of the work. Kim Tron's a longtime technician in my lab. Jill and Andrew are former grad students who did a lot of the cloning. This work is actually part of a program that we have with Mark Asun, who will be talking later, and Octavio Ramillo at the Nationwide Children's, and Stefan Nivex and General Lee at Ohio State. Thank you.
Do you have any idea what sort of the steps, or the step that is influenced by the NS1 in the virus replication cycle?
Well, so I think it's complicated. So what we can find, so when we look at the NS1 deletion viruses and even these single amino acid deletions, we see that there's a significantly lower amount of genome or anti-genome length RNA. But the transcription levels are relatively similar for the viruses. So we think it might be something to do with the replicase and not transcriptase activities.
Any other question? Well, if not, thank you very much, Michael.